Hi, I'm Josh Steele, and I'm from the Southern California Coastal Water Research Project, and I'm going to present some of our work on antimicrobial resistance in effluent streams that discharge into coastal waters. So there's been a lot of news lately, some of it from based off of EPA research that said that not only are there superbugs in sewers, but it's possible that they're actually being concentrated or selected for and increased throughout sewage treatment plants, which would cause a public health issue when that sewage effluent is discharged. And so we wanted to investigate whether or not the wastewater treatment plants in Southern California discharge antibiotic-resistant bacteria or antibiotic-resistant genes, and is there any difference in the treatment levels of these sewage treatment plants? And then our next question we had, ideally once we find the answer to that, was can we detect sewage bacteria or antibiotic resistance genes at a beach near a sewage treatment outfall? So the way we went about this, our approach was to get as many different treatment plants as we could in Southern California, and we got 10 with treatment that ranged from primary all the way to tertiary treatment, so very limited treatment all the way to very, very high level recycled water treatment. And we looked with a few different methods for viable bacteria, focusing on high priority antibiotic resistant bacteria like carbapenem resistant Enterobacteriaceae, vancomycin resistant Enterococci, methicillin resistant Staph aureus. Then we also used molecular methods to look at antibiotic resistant genes using digital PCR, qPCR, and sequencing. And finally, we also wanted to see if we could find a sewage signature using the microbial community sequences themselves. So let's see what we found. When we looked at the viable bacteria, we looked at both the bacteria coming into the sewage plant, the influent, and coming out in the effluent. And what we found is there was a reduction in nearly every case in the groups, the high priority groups we looked at. So for the carbapenem resistant bacteria, we found a 20% reduction, or 80% in the effluent was in the primary. Once you got to secondary and tertiary, that was reduced down to less than 1%. In contrast, with the methicillin resistant in primary, we saw very little reduction. It was nearly identical what was going into the plant and coming out of the plant. But again, once you got to secondary and tertiary treatment, particularly tertiary, there was a strong reduction. When we looked at genes, and these are the carbapenemase gene copies, we found that there was still a reduction, but it was not quite the same pattern as we saw in the viable bacteria. So now these may be viable or non-viable bacteria. These are just the genes themselves, and which is why I'm showing you concentrations. And you can see in the influent, which is the gray bars, that we had quite a few of the carbapenemase genes coming into the plants, and they were reduced in almost every case. And particularly once we got to tertiary treatment, and especially the highest level of tertiary treatment over here, they were nearly eliminated. That was not the case when we looked at a broad set of genes, that they were all eliminated. We still saw fewer in the tertiary treatment plants, but we did find genes in every single effluent that we looked in. So what about a sewage signature? So now we know we have antibiotic resistant genes coming out of the plants, but can we track this? Can we trace this once it actually comes from the effluent into the ocean or into the recreational waters that it's discharging into? Well, we looked at the whole community, both in the sewage coming into the plants and the sewage going out of the plants, the treated effluent going out of the plants, I should say. And this is a lot to look at, and in the interest of saving time, all I want to point out is that the influent is very similar across Southern California. We see a lot of the same groups in the influent, in the microbial community, coming in. But if you look over effluent, you can see that each plant, shown here in the brackets, these 4 little bars of each one, has a different signature. They almost have a unique plant-specific signature. This makes some sense. All these plants are sort of one-offs, and their treatment trains are unique, but what's coming out of them is a unique community. And we think that this is a way we can track the sewage once it comes out of the plant, since this is the effluent that's being discharged. So we looked for a place where we could do this. And one of the projects we had in collaboration with EPA and EPA funded was to look at sewage and antibiotic resistance genes um, across the US-Mexico border in San Diego. And you can see here on the map, we have a treatment outfall um, down here in Mexico, and it actually is a beach discharge, not an offshore ocean outfall. And when there's a south swell, there has been evidence that the plume from that effluent discharge makes it all the way up to San Diego to the beaches here. So we went out to test this in collaboration with partners across the border and the cities in San County and San Diego. And what did we find? Could we see a sewage community? In fact, we saw a very strong sewage community signal. So this is a bit of a complicated graphic, and I'm going to walk you through it. But what you have on the top here, and each of these is a different day that we went and tested, is from these sites that you saw before, from south to north along the bottom. You have the ocean community signature on the top, and you have the sewage community signature on the bottom. The ocean community signature has the bacteria you'd expect to see, Synechococcus, SAR11, the marine group Flavobacteria, etc. And the wastewater treatment plant has the bacteria you'd expect to see, Archobacter, Aeromonas, Comomonas, what have you. But what you can see is that we not only detect a very strong sewage signal right at the outfall here, but we can detect it all the way up the beach when there's a south swell, when there's a current pushing that up and all the way up into San Diego until we get to the very northernmost part, and then we sort of start losing that signal. So can we use that microbial community signature to track the sewage up current and up the coast? Yes. Now, what about the antibiotic resistance? Well, using the same samples, and this is the same south to north gradient, we have two different antibiotic resistance genes we took a look at. And you can see here again is the sewage outfall at the beach. And these are during the same day 1, 2, 3. And you can see that we have a similar gradient. In this case, some of the genes reach all the way up to that site where we couldn't detect that signal. So we're not only seeing a sewage community making it all the way up to San Diego and Tijuana and beaches that people use, we also see antibiotic resistant genes making it up current. So this is all very interesting. And the question is, okay, well, we're seeing the genes, we're seeing the signal, but people aren't really getting sick. So where are these genes going? What are they doing? If it's not making people sick when they go to the beach, what are these genes doing? Where are they going? One of our thoughts was that maybe the genes are going into natural bacterial communities and sort of hanging out there. So we took a look at, in partnership with some of our collaborators at UC San Diego, Rachel Diner in particular, and you should go see her talk for more details on the Vibrio isolates and Vibrio communities. But we took a look at the antibiotic resistance genes in these communities and tried to see, well, could we match some of these with what we were seeing in the sewage and what we were seeing in that effluent plume? And in fact, we could. And what you see here, we have carbapenems in red and other penem resistance, along with other macrolides and some of the other genes we saw that are showing up in Vibrio isolates isolated from estuaries in Southern California. This is an indirect comparison, but it does show that there may be an influence of some of these effluent genes going into the environment, even if it's not showing up in a public health response where people are getting sick at the beach. Now, this could be a reservoir that then will affect people in the future or possibly affect animals or other potential environmental health issues. So in quick summary, we can not only see reduced but not eliminated resistance genes in sewage, but we can track that sewage and track resistance genes into the beach and ocean waters in Southern California. And with that, I just want to acknowledge all the people who assisted in this work, the sanitation districts, City of San Diego, County of San Diego, Imperial Beach Lifeguards, US EPA, Zymo Research, Wild Coast and Proyecto Fronterizo Educación Ambiental, and Surfrider Foundation. Thank you.